It's great to be here. When Bruce called me up a few months ago to ask if I would give this lecture, of course, I immediately said yes. But then I became a little bit terrified. First of all, of course, you can tell there's no pressure, right, Bruce? But secondly, obviously, you're coming back to a place that you really grew up in, and so you obviously want to do well. And I have such enormous respect, both for the laboratory and in particular this meeting. As Bruce said, this symposium really brings people together from all over the world to discuss advances in science. It also supports, of course, the research of many fantastic scientists here. And I think the story that I'll tell you is sort of an example of the type of research in science that can come from sort of discovery-based science, curiosity-driven science that isn't intended initially to discover something important, but eventually ultimately does. And so there's a lot of excitement about the biology. We're not there, and I'll try to tell you as far as we're at. But again, I think this is really a great example of how science can work when we work together. So how many of you ever heard of zombie cells? Just could I get a show of hands? That's what I'm going to talk about today. This comes up in the news every now and again. Okay. I know some of you are a little shy. But I want to tell you about the zombie cell and how it may play a role in human biology and human pathology. And I just want to start out kind of with giving you the highlights of what I want you to think about during the course of this lecture. So zombie cells are a cell state with striking features that were identified serendipitously. No one was looking for these cells, but they were found. This state is now known to have very powerful biology in influencing tissue repair, cancer, and aging. It's a very broad scientific ramifications. Studying the impact of this state on tissue biology is helping decode disease mechanisms across a wide range of pathologies. And it's pointing to a new class of therapies with very broad-reaching therapeutic potential. Okay, well, what about zombie cells? This has been a term that has been coined in the popular press. This is an AI-generated diagram that describes the excitement of zombie cells, cells that are around that contribute to disease. And by eliminating them, we can improve various disease. So what are zombies? I guess we probably all know these are sort of described as the undead, supernatural beings that are dead, but are not dead, but they're not really alive. But zombie cells have been in the news. And again, if you haven't seen it, I'll just show you a few headlines of why people are sort of excited and interested and fascinating with zombie cells. In the Washington Post, here's a headline that zombie cells may hold the clues to improving old age. In The Guardian, so this is not just a US-specific thing, in the UK they're excited as well. If you want to live forever, you flesh out these zombie cells. National Geographic pointed out research that linked zombie cells to Alzheimer's disease and suggested that they might hold the secret to a cure. And in other publications and news releases, again, sort of a disturbing thing, zombies, are they already in your head? Do we have to worry about these? But not everybody has reported bad things about zombie cells. Some reports suggest maybe they're not so bad, maybe they're harmful, helpful. Despite their scary name, maybe they're not all so bad. But really, I think where the news media has come down is that we don't want these cells. To stay young, kill zombie cells. And in this particular one, really the extreme. Let's just go kill zombie cells, okay? We got to get rid of these cells. So what are they? That this is all this fuss is about and this sort of news and the sort of name zombie cells. Well, the term zombie cells relates to senescent cells. And I'm going to tell you what these cells are. But the meaning of the word senescence, this is from the dictionary, is a condition or process of deterioration with age. And so these cells were initially linked to age or an age-related or a time-dependent decline in cell function. And it goes all the way back to the early 1960s with a guy named Leonard Hayflick, who originally described zombie cells. What Leonard Hayflick was trying to do in those days was to take cells out of people and put them in a petri dish so we could study them as an experimental model. And it was known that if you took cancer cells from people and put them in culture, they would grow forever. But if you took normal cells, and in particular they took skin cells, they didn't. So here's a picture of Leonard Hayflick. Unfortunately, he passed away late last year. And what he did was to take cells and put them into culture and then count how many times they would divide over time. And so here's a graph of what he saw. Initially, the cells started to proliferate, but eventually they stopped dividing. They didn't double anymore. And that process ultimately was described as senescence, the fact that these cells have a replicative exhaustion in culture. Now, of course, these are cells in culture, but it created a fascination about what this failure or this loss of proliferative capacity over time might mean. Some people thought perhaps this was telling us about mechanisms that prevent cancer. Of course, cancer cells divide forever. These cells didn't. Maybe they were activating a process that was important to prevent cancer. Other people said this might be reflective of aging, right? As we know, as we age, our cells decline, and maybe this phenomenon was because the cells got old. Now, these zombie cells, as they become now described, have very interesting features. If you just look at them under a microscope, these are normal cells, fibroblasts, how they look. And the zombie cells don't look as good. You can see they're flat. They sometimes, I guess, in the field, we often say they look like fried eggs. But in any case, they've changed the way they look. They've stopped dividing. They look funny. And here, I think, is a good example of why they're called zombie cells. These are normal cells that divide, and you can see them proliferate here and eventually expand in the petri dish. But these are the senescent cells, the so-called zombie cells. They're just sort of sitting there doing nothing. They don't divide. They don't die. They're just there. They're present. Now, over the years, people have identified markers, features of zombie cells, senescent cells, that they've used to characterize them. Some really were identified serendipitously. One of the ones that's most widely used was discovered by Judy Campisi, who's really has been a dominant feature in the field of cellular senescence. Again, unfortunately, she passed away late last year. And notice that if you stain cells a certain way, the senescent cells turn blue, whereas the normal cells don't. This marker, SA-beta-gal, has been widely used to identify senescent cells, even though the basis or the relevance of this to the program is still incompletely understood. Another factor that's been linked to senescent cells really had its origins here at Cold Spring Harbor Laboratory. Work from Manuel Serrano, who's probably here in the audience, together with Greg Hannon and David Beach, found a molecule that they showed regulated cell proliferation. It was called P16. When this molecule was present, cells didn't divide. And interestingly, this molecule was lost in cancer cells. That makes sense. Cancer cells divide uncontrollably. They lose P16. But then later, they, together with other colleagues, showed that P16 went up when cells underwent senescence. And this is just looking at extracts of cells, either growing or senescent, different cell types in culture. And this measures, this intensity is the abundance of this P16 protein, and you can see that it goes up. So senescent cells look funny, they stop dividing, they get this marker SA-beta-galactosidase, they turn on P16, which maybe helps them stop dividing. So in the end, what causes Hayflick senescence? Remember, these are cells taken out of a person, put in culture, they grow for a while, and then they stop, as shown here. Well, it turns out, work from a number of labs, but really dominated a lot by science here, identified that as cells divide, their chromosomes get shorter. And that's because they don't express an enzyme called telomerase. Now, this enzyme was discovered by Elizabeth Blackburn and Carol Greider. Carol Greider, of course, worked here for many years and is one of the Nobel laureates that I will aspire to try to be. But no, that preceded me here. But in any case, what they found is that when cells turn on telomerase, which happens in cancer cells, they can continue to extend their telomeres. So what happens is cells divide, their telomeres get short. All of a sudden, it causes a problem, and the cells undergo this senescence program. So that's what happens in these cells in a petri dish. But is this really relevant to aging or cancer? Well, we don't know for sure, but there are hints. And so one hint is that in human cancers, about 90% of them turn on this telomerase that seems like they need it to become a cancer, to be immortal. And about a quarter of them actually have a mutation that's associated with the cancer and not normal cells that turns on this gene. So telomerase is associated with cancer. Now, Carol Greider, together with Rhonda Pino, knocked out from a mouse the telomerase gene. And interestingly, what happened to these mice is they prematurely aged. Here's shown on the right compared to the left, suggesting again that telomerase somehow protects us against aging. Now, could this be related to senescence? Maybe, but we really don't know. It could be true, and unrelated. Now, another experiment here that was informative, and this was the one that was very important in my career, that I did together with Manuel Serrano and David Beach here at Cold Spring Harbor, was a different way of looking and seeing senescence. So young cells divide just fine, but what we found is that if we introduced a cancer-causing mutation, a gene called RAS, into these cells, not only did they not become cancer, they stopped dividing altogether. You can see that just looking at the division of the cells shown here. And if you disrupted the p53 or the p16 gene, these cells went on to grow indefinitely. Now, RAS was actually identified here, as well as at MIT, as one of the first genes that were ever identified in human cancer that could cause cancer. Yet here we see something that seems to be precisely the opposite. In fact, if we look at the cells, we can see that these cells have this senescence-associated beta-galactosidase, suggesting that they were senescent. And so this led to a model. Again, it's a model, it's not proof, but it says that maybe cells that acquire cancer-promoting mutations don't become cancerous with that mutation alone because they engage senescence, which would stop that from happening. This process involves p16, which we know is involved in senescence, another important gene, p53, that are inactivated in cancer. So maybe the way you become a cancer is you fail to undergo this senescence program. And it sort of suggests that all of our cells have a safety valve that prevents cancer from coming. And that sort of has to break for cancers to develop. And again, this is an interesting idea. It's a model. But there's some data to suggest it might be true. This is work from Daniel Paper. There were a whole series now already in 2005 of papers. Many of the people are here in this audience that published them that suggested you could actually find senescent cells in precancerous lesions. Now, we all have moles, right? Everybody has one. They sit in our skin, usually forever, and nothing happens. Every single one of them has a cancer-promoting mutation, yet they don't give rise to cancer. And what Daniel showed is that those cells had features that look senescent, which was consistent with the idea that this process prevents cancers from developing. So that says the zombie cells are pretty good, right? They help protect us from getting cancer, but that's not where the story ends. Turns out, if you look in other tissues, other pathologies, COPD, MASH, diabetes, osteoarthritis, Alzheimer's disease, you can find that these cells accumulate. This is done by looking for the molecule p16. You can see this in tissues in pulmonary fibrosis. Here's looking for this SA beta gal marker in liver fibrosis or MASH. And so we start to see these cells accumulating. They also accumulate when we age in all of our aged tissues. So that says, well, maybe they're not so good after all, but of course, they're just sitting there. We don't know what they do. But then there were some functional studies, again, many of these are done in animal models, This one was done by Ned Sharpless, who's the former director of the NCI, where he engineered mice, I'm not going to go into the details, that could become fluorescent if they turned on this P16 protein, right? That's the marker that goes up when cells become senescent. And you can see if you have young animals, they're not really fluorescent. But if they're old, they accumulate this protein, suggesting that these cells accumulate when we get old. He also went and took blood cells from people at different ages and tried to measure the levels of P16. And you can see, if you just look at chronological age, roughly, it's not black and white, as we get older, the cells in our body express more P16, which is consistent with us accumulating senescent cells. But here's the big experiment. Darren Baker, together with Jan van Derzen at the Mayo Clinic, generated a system where they could take advantage of cells that turned on P16 and were presumably senescent, and then add a drug that killed just those cells. They waited for mice to get old. They killed the P16 cells. This is what the mice look like. This is an old mouse. This is the same age where they eliminated the senescent cells. So that is where people got excited in the popular press. It said that these senescent cells might drive pathology, age-related pathology. And in fact, these sort of experiments were done in all kinds of other things, fibrosis, osteoarthritis, where similar effects were observed. So that says maybe if you get rid of these senescent cells, you can affect human disease. And again, that led to sort of super excitement, maybe over-exaggeration of what the potential of these so-called senolytics is, but that perhaps, this would be a treatment for age-related tissue decline. Well, we're not there yet at all, but it is sort of interesting and exciting biology that drove the field forward to try to figure out more what was going on, how this process would work, and could you ever manipulate it therapeutically. And so nowadays, we sort of think as senescent cells of having these two opposing effects that we still don't fully understand. On one hand, perhaps they're beneficial. They prevent ourselves from getting cancer. But on the other hand, if too many of them accumulate, it leads to organ decline and tissue dysfunction. And so again, we have these two zombies, the bad zombie and the good zombie, and we need to figure out why they're different and what they do. So there's been a lot of work on trying to understand what are the features of these senescent cells. What is it about them that makes them so special or unique? And a lot of that work has been done on those cells that Hayflick initially identified. And so when one characterizes these cells, again, many people here in this room have been involved in this work, we've learned that they have features that differentiate them from other normal cells. And I can sort of divide it down into two major sort of processes that we think is going on. One is the cells stop dividing. That's what Hayflick initially described. That involves molecules we know are important in cancer. It turns off genes that are needed for proliferation. But then the other one is sort of more interesting in a way. The cells secrete factors that help them communicate with other cells in the tissue. This is a process Judy Campisi initially called the senescence-associated secretory phenotype. And it suggests that senescent cells, when they're present, they can do things to the tissue. And these features of the SASP, again, I won't go through all of them, but it's hundreds of different factors are released from these cells. They can be different depending on which type of cell is senescent. I think that's an important clue. They can alter immune function, and probably the fact that different senescent cells release different factors explains these different effects, or at least contributes to these different effects of biology. Now, one of the things that my lab has studied is when senescent cells release these factors, sometimes, we don't always know why, they can recruit the immune system to kill the senescent cells. And here's a movie that shows that. So what you're looking at here are the green cells, are cells in culture. These are proliferating, these are senescent. And these are senescent, but we blocked this secretory program. The red cells are immune cells. They're called natural killer cells that can attack cells that are stressed. And so what you see in the movie is if you look here versus here, that in this setting, when the cells are senescent, the red cells are attacking the senescent cells. You see that they die. And if you disrupt this secretory program, that happens less well, suggesting that this secretory program is needed for the senescent cells to target the state. You can see that in tissue. This happens to be in the liver, where the brown cells are cells that are triggered to undergo senescence. If we look over time, if the SASP is present, the secretory program, you can see they go away. But if it's blocked, they persist. And here you can actually see these small purple cells are the immune cells attacking the senescent cells when the SASP is on. But again, they don't see them at all when SASP is blocked. And so this has sort of led to an interesting idea of how senescent cells could be good or bad. If they're there for a while and then they get eliminated, maybe that leads to tumor suppression and tissue rejuvenation. But if they don't get eliminated, they could accumulate and cause a problem. Interestingly, as we get old, our immune system isn't as good. And so it's an interesting model, not proven, that perhaps one reason senescent cells accumulate as we age is our immune system isn't as good at eliminating them. So there's a lot of interesting questions in senescence research. We want to know what these molecular features are and what would distinguish what would be good and bad senescence, if you will. How do these cells impact the tissue state and function? And when and how would they be cleared by the immune system? If that's the difference between good and bad, we'd like to understand it so we could manipulate it. And of course, all of these experiments I've shown you are sort of in artificial systems. Could you actually therapeutically manipulate this, and what would happen? Now, one of the challenges that we faced is that all of the properties we've studied, or many of them, were defined based on putting the cells in culture. And so this is, of course, an artificial circumstance. And just to give you an analogy of the potential problems or challenges this encounters is if one were to study Atlantic cod and define a definition of Atlantic cod based on it being out of an organism, this is what it might look like. You would say Atlantic cod's motionless. It's brown. It's dry sometimes, and it's hot. But of course, Atlantic cod isn't at all like that in a real fish. It moves, it's multicolored, it's wet, it's cool. And so again, the definitions that we have of senescence might be biased by the fact that we studied them in an artificial context. And so to really understand the powerful biology of this process, we need to study them in real tissue. Now, it's driven by new technologies called single-cell technologies that are extraordinarily powerful. This is an analogy that many of the scientists in the audience will have seen, but I think it's informative to think about how these sort of technologies work. So if this is a tissue, and the fruit are different cell types in the tissue, they're organized in a certain way, there's different numbers of each fruit. Now, the way we typically, until recently, analyze tissue is to take a biopsy, right? Which, of course, is a blend of all of these different cell types. You're going to get a mush. Maybe if there's more strawberries, it'll be redder. Maybe if there's more blueberries, it'll be bluer. But nevertheless, we're lacking information. Now, one can now separate the individual cells and characterize them molecularly and look at them in each detail. And this will tell you exactly what cells are there and how many. But even more recent and powerful and expensive technology is called spatial profiling, where it really puts the cells back in each one and understands the molecular details of each one, who they're next to, who they might be talking to, and so on and so forth. I'm going to give you an example from our lab of how this can work. This happens to be from the pre-malignant pancreas, so mice that are predisposed to pancreas cancer, where we can isolate single cells from these mice. You can go through this process called single-cell RNA sequencing. And then because you get so much data, really billions and billions of data spots, there has to be ways in which it's presented so our brains can actually understand it. And these are called dimension reduction examples. And I'm just going to show you through how this actually works. And hopefully it'll be simple. So if you have different conditions, we're color coding them here, and you have an individual dot as a cell, this cell has certain states that, again, are all sort of reduced down into this single spot. If the cells are next to each other, the way this is represented, they're similar to each other. And if they're far apart, they're very different. And by taking advantage of what we know about different cell types, we can actually define which cell states are present in this tissue and characterize them based on the color codes here. Now, this turns out to be really powerful, and when you do this sort of approach on premalignant pancreas, you can see that there's a subset of cells we're calling the blue ones that have features of senescence. They look like a progenitor cell, a stem-like cell. They have other features, but they're not dividing. They express P16 and P53, and they have factors that look like this SASP. So these may be in a tissue, the cells that are actively undergoing oncogene-induced senescence. Now, the spatial methods, as I said, are even more powerful. You can take these tissues and put them on a slide and stain them with thousands of probes that allow us to identify almost every single molecule in every single cell and then look at where it is spatially. You can imagine this creates a ridiculous amount of data, enormous amount of computing power, literally billions and billions of data points, and you need to have computational biologists to deconvolute it. But what ultimately comes out is a tissue that has different color codes based on state, and each one of these spots really has about 20,000 bits of information. So again, we only can show for our brains to understand them some. When you do this again, you can see sort of the richness of tissues color-coded by different ones. These ones here and these ones here have features that we might imagine as senescence. They don't look at all like Hayflick cells, but they're close enough that we think they might be senescent. The other thing about the spatial technology is you can start to look at where these cells and who they're around. So here in blue are patches of what we believe to be the senescent cells in a tissue. And you'll notice certain cells that have fibroblasts, these are called, in yellow, certain immune cells, macrophages, in green, surround these senescent cells. So it's a very different environment where these senescent cells are compared to the rest of the tissue. This is really interesting. Because, and here's a senescence marker, it starts to tell us how the senescent cells might affect its surroundings. Now again, senescent cells release factors, they're often called ligands. Surrounding cells have receptors, and if you start to see expression of these in adjacent cells, you start to infer that they're talking to each other. And this is really powerful because these are the ways in which the senescent cell executes its biology, and they provide possibilities for therapeutic intervention. So how can we trigger senolysis therapeutically? Of course, this is what we want to do, particularly in aged and damaged tissues. And there's been a lot of discussion on this meeting of molecules. We call them senolytics. They lyse senescent cells. And there's a whole series of them that are in clinical development, and many of them are in clinical trials. And James Kirkland, who spoke earlier, is really a pioneer in trying to reduce these into clinical practice. All of these are small molecules. These are typical molecules like a cancer drug or aspirin or anything else that sort of go throughout your body, titral cells, and their activity is dependent on the fact they affect the disease state differently than the normal state. Many of these, again, are identified empirically. We don't always know why they work, and it's difficult to predict in advance when they'll work and when they won't. That's one of the things we'd like to have are biomarkers that say this patient should get this drug. They can have substantial toxicities. And of course, for things like aging or tissue decline, safety is really important. We can't be giving toxic drugs to these patients. So another approach, which we've really worked on in my laboratory, together with others as well, is to take a different approach, to target proteins that are expressed on the outside of a cell, on the cell surface. These can be recognized by antibodies or other therapeutic approaches. The thing about this that's nice is that you know which cells express the target. So these are the cells that should be the ones you eliminate or get rid of. And you know whether there should be toxicities, because if normal cells express that protein, that could lead to their elimination as well. So you're looking for proteins that are on the disease state, not on the undiseased state. And while this is widely used in cancer, some of the major anti-cancer drugs work through this mechanism. They hadn't been used very much in senescent cells. One of the molecules that goes up in senescent cells is a molecule called UPAR. This is a molecule that normally is involved in wound healing. It's not usually expressed on normal cells, but it is induced in senescent states. Turns out there's a part of it that gets released in the blood. It's pretty cool, actually. And you can measure it using a diagnostic test. And it's actually been used to measure the disease severity of a whole series of different indications. Again, keeping in mind this might mark senescent cells. If it goes up, there might be more senescent cells. That would be bad. And interestingly, if we look in aged individuals, people, it's higher. It's also higher in aged mice. So this is a marker, perhaps, of the number of senescent cells in a body. And if we stain for this marker across different pathologies, they're there. So again, consistent with senescent cells, you see a lot of them here. Now, there's exciting new methodology that was really pioneered by Karina Moore when she was in my laboratory. She's now here at Cold Spring Harbor, where she's carried on this work in her own lab, which is using CAR T cells. This is a therapy that's been used to treat cancers by targeting cell surface proteins specific to cancer. And Karina's idea was, could you use them to target senescent cells and make them a senolytic? And she did that. And I won't go through all the data, but there was 2 really fantastic publications that she did. And I'll just show you one example of what happens. This is the image of a fibrotic liver. This is an image of the liver that's been treated with these CAR T cells that eliminates senescent cells. She's also went on to treat old mice to see what would happen. And it turns out they're not toxic, these CAR T cells, but they do reduce inflammation. They improve the way the mice exercise. They have better endurance. And they have less metabolic syndrome. Their insulin responses are much better, more like young mice. So this is a potentially interesting therapeutic approach. Another molecule that I actually talked a little bit about earlier in my presentation today is called P-Select, and this is also up in senescent states. And it has interesting features in that it binds a molecule called fucoidin. This is sort of a complex sugar that you find in seaweed. And what's interesting about that is my colleague at Sloan Kettering, Dan Heller, has shown you can package small molecule drugs in fucoidin. And this is sort of a cartoon of how it looks. One of the drugs that's a known senolytic, kills senescent cells, can be packaged in this drug, in this molecule, where the presence of fucoidin makes it specifically go to the cells that have P-selectin, the senescent cells. The advantage of this, of course, is that it might be less toxic because it's not going everywhere. It's only going to the senescent cells. You can actually see that here. These are cells in green that have this protein fucoidin. Some are endothelial cells, and these red cells are the ones that are senescent. The white ones are the ones that take up the particle, and shown in yellow are the ones where they... So the endothelial cells, which you wouldn't want to get, don't get targeted, but the senescent cells do. Now, if you have the senolytic in these drugs and you try to treat senescent cells, on the left you're going to look at cells that are not senescent. On the right, the ones that are senescent. And if they turn green, they die. So if we give them the nanoparticles, you'll see on the right that these cells are killed. Just like we saw with the CAR T cells, this is an example of mice that have liver fibrosis. This red here is the fibrosis. And you can see with these nanoparticles, it's reduced. Not quite to the normal liver, but it declines. And if these yellow cells here are the senescent cells, they go away. So there's true senolysis. But what's particularly powerful about this point is navitoclax is a drug. It was in development by AbbVie, and it's just too toxic for people to use. So it's actually been pulled off clinical trials because you get bleeding disorders. But it turns out, at least in animal models, in the nanoparticles that doesn't happen. They're not toxic. So this is just measuring bleeding time. In mice that get the free drug, you can see compared to normals, it takes longer. It takes longer to clot. But these nanoparticles, they are just like normal. So we get the activity of the drug without the toxicity, suggesting this be a safer approach. Now, just the last couple of slides I'm going to show you really comes back to the intersection between senescence and cancer. We know this is a process that's interrelated. Senescence, for example, cancer is, of course, a disease of aging. In one of the areas, there's a lot of ways in which we can exploit this therapeutically, but I'm just going to talk about one, which relates to the problem of cancer immunotherapy. Work initially from James Allison and many others won the Nobel Prize for developing drugs that convinced the immune system to reject a cancer, just like they'd reject a cell that had a virus. These are so-called immunotherapies, and they cure patients, but only a fraction. About 12% of patients benefit from these, but the other ones don't, and people have wondered why and how to make that better. And so one of the things we've wondered in my lab and other colleagues here at the meeting is whether you could ever use what we've learned about senescence to actually improve immunotherapy. So one way would be to sort of target the bad senescence. Remember, I told you that tumors are often fibrotic, senescent cells accumulate in fibrotic regions, and this actually has been thought to sort of prevent the immune system from getting in and rejecting the tumor. If we use these nanoparticles that target senescent cells in mice in combination with chemotherapy, we see some interesting things. So here in the green color is a tumor. These are mice that have a liver cancer in their liver, and if we treat them with immunotherapy, this tumor shrinks. But if these mice have fibrosis in the liver, where all these senescent cells are accumulating, now that therapy doesn't work. But if they have fibrosis and we give them these nanoparticles that kill the senescent cells, now you can see we get a good response to therapy. And this is what it looks like in terms of survival. The bottom is really the thing. Without the nanoparticle, 100% of the mice eventually succumb to disease. With the nanoparticle, 80% of them have a complete response. So again, this suggests that we can eliminate senescent cells in fibrotic tumors to improve existing immunotherapies. The last way I'll show you is inducing good senescence, like we see that protects against cancer, to promote immune surveillance. Remember, I told you sometimes senescent cells get seen by the immune system. Maybe if we can trigger that state in cancers, the cancers will get seen by the immune system as well. If you trigger senescence in tumors, they can sometimes respond to immunotherapy, and so we need to understand precisely how that works. Anyway, so just the take-home messages. The term senescence describes a very diverse set of cell states that remain incompletely understood, but they have very powerful biology. And these cells, or at least that have features of senescence, can contribute to a broad range of pathologies. And preclinical studies indicate that manipulation of senescence can have exciting, almost magical therapeutic properties. And so therapies that target senescence, which accumulates in a whole range of diseases, have the potential to transcend disease states. Such as one drug that might be effective against fibrosis in the liver might also be effective in Alzheimer's patients. Okay. That's the lesson from preclinical models, but there's the cautionary notes. If this was a commercial, all of this would be going very fast in the background with some happy little picture. But the bottom line is, this is very exciting biology, but I don't want you to go home thinking that everything is solved. Experiments in preclinical models do not always translate to people. The biology seems to be exciting. We've correlated it with people, but again, we don't yet know whether we'll get the same sorts of results. Of course, treatments in frail and aged populations must be safe, and that will be an important driver of determining how and when we can bring these sorts of therapies to different sort of indications. And so what this really all means is that we really have to have a better understanding of senescent cells and their normal biology if we want to really exploit that therapeutically. We started out with Hayflick cells identified in 1961, and over the years learned an enormous amount about how these cells work and what they might do, which has led to some exciting possibilities, but we're not yet there. And that is the reason why we need to continue to support fundamental research into these sorts of questions. And so again, the goal is taming the zombies. And I want to thank you for your attention, and I'll stop and take any questions.